Hi, my name is Pranay. In this talk, I will give you an introduction to Dynamic Causal Modeling or DCM. I will explain the principles underlying DCM, how it is used to model MEEG data, and what kind of inferences can be made about the brain using DCM. Typically, in an MEG or EEG experiment, inference is made on average time courses or ERPs or measures in the spectral or time frequency domain with the ultimate goal of inferring how activity inside the brain could be explaining and accounting for sensor level data. Now there are various ways of modeling how regions in the brain interact and produce sensor level activity. One of the ways is structural connectivity, which relies on our knowledge of anatomical connections between regions. The other is functional connectivity, which models statistical dependencies between time series observed at different regions in the brain. What DCM does is it models the effective connectivity between regions inside the brain, which means that it captures causal directed influences between neuronal populations and builds upon our knowledge of both structural as well as statistical dependencies between various regions. DCM is a Bayesian framework for estimating this effective connectivity from observed data features. It consists of a generative model of interacting neuronal populations, which are modeled as coupled dynamical systems that evolve over time. This framework allows us to carry out hypothesis testing on the connectivity as well as perform inference on the estimated parameters of the generative model. I'm going to start with describing the generative modeling approach in DCM. I'll then proceed to laying out the landscape and explain the different kinds of DCM approaches available. Following which I'll move on to neuronal models and develop an understanding of the Jansen-Rit ERP model. Following this, I'll come to observation models and model inversion, and then end with a summary. The DCM generative model consists of a neuronal model and an observation model. The neuronal model consists of neural state equations which describe dynamics of brain activity. These state equations are a function of the current state of the system, an input which is u, and theta 1, which is a set of variables which parameterize the state equations. These state equations describe how different populations within a patch of cortex interact with each other and result in an output. This output is essentially the depolarization of pyramidal cells in layer 5, layer 6. It is this depolarization happening in the dendrites of hundreds or thousands of pyramidal neurons in a patch of cortex, which ends up producing an electromagnetic field which is strong enough to produce activity at the scalp which is picked up by sensors. The observation model essentially takes this brain activity at each source in the brain and maps it to observed data features. The observation model consists of a forward model, which could be a lead field in the case of EEG-MEG, and consists of a head model with various layers and their respective conductivities. or it could be a gain function in the case of recordings of local field potential. Ultimately, the observation model translates the activity of dendritic depolarization in a patch of cortex to activity that is observed at the scalp or on sensors for EEG or MEG. Taken as a whole, the generative model consists of neural state equations which define how different regions are connected to each other and how dynamics within each region evolve in each of its populations. And the observation model takes the output of each of these region, which is the depolarization in its pyramidal layer, and maps it onto observable measurements at the sensors. The observed data features could be in the time domain, like event-related potentials or fields, or they could be frequency domain features like power spectra. And we usually have a hypothesis about which regions in the brain are responsible for generating these data features, as well as how the regions might be connected. Essentially an architecture underlying the observed data features. DCM gives us a way to link the hypothesized mechanisms with the observed data features, allowing us to ask the question, what measurements of brain activity does our model predict given some parameters? Parameters here could be some variable that captures the effect of an experimental condition on a specific connection, or parameter could be a specific synaptic property of a population. Conversely, and what we are usually trying to achieve is we are trying to estimate a model based on our hypothesis and some observed data. Here we ask the question, what parameters of the model best explain observed measurements? We then carry out inference on these parameters after fitting the generative model and carry out hypothesis testing, in which we ask whether an experimental input affects a specific connection or whether the experimental condition modulates a specific synaptic parameter. In DCM for evoked responses, we are primarily trying to model event-related potentials or fields which are observed at the sensor level, and we have hypotheses about the underlying neuronal architecture responsible for generating these ERPs and ERFs. This is what I'm primarily going to discuss in this talk, but before I do so, I would like to briefly touch upon the various kinds of DCMs out there. Broadly speaking, there are two kinds of DCMs. One, physiological DCMs, which consists of biologically plausible underlying neuronal models, and which can generate a richer set of dynamics. And the other is phenomenological models, which have simpler underlying neural models. The physiological models model sensor level data, whereas the phenomenological ones only model source level data, which means that the inverse problem is included as a part of the observer in physiological DCMs, but not in phenomenological DCMs. Because of this, the physiological DCMs also optimize location and orientation of sources, whereas in phenomenological DCMs, locations are specified and are not optimized. Lastly, physiological DCMs can test for number of sources. So I could compare a model with 4 sources versus 3 sources and examine their model evidence to pick a winning model, but I cannot do that with a phenomenological model. The physiological DCMs consists of DCMs for evoked responses or DCM-ERP and DCM for cross-spectral densities or DCM-CSD. Both of these are modeled using rich neural models, which can either be neural mass models or field models, and they describe the dynamics of various populations in a cortical column as it unfolds over time in response to an input. The input for the ERP is a Gaussian pulse, whereas it is Gaussian noise for CSD. I will explain physiological DCMs in more detail and touch upon the kinds of questions we can ask using DCM-ERP. The phenomenological DCMs consists of DCM for fMRI, which is the subject of another talk in this series. DCM for induced responses model changes in power caused by external input and or coupling with other regions. For example, beta activity in region A leads to gamma increase in region B. And you can ask the questions, what regions are connected? Or does slow activity in one region affect fast activity in another? Finally, we have DCM for phase coupling, which models synchronization and entrainment through phase coupling between regions. This allows us to ask questions like which regions have a master-slave relationship, and it also allows us to perform inference on frequency-dependent coupling values. For the rest of the talk, I will be focusing on DCM for evoked responses, where we will model observed event-related potentials or fields on EEG-MEG using a generative model. I'll first begin with the neuronal models. The earliest attempts to model EEG phenomena involved various approaches to model generators of the cortical algorithm. There were existing models of excitatory inhibitory balances in the thalamus and other areas, and Lopez da Silva in 1974 used an existing model developed by Wilson Cohen and Walter Freeman for modeling alpha activity generated in the thalamus of a dog. They used a model which consisted of thalamic relay cells, a couple of hundred of them, and inhibitory interneurons. The relay cells fed excitatory projections to the inhibitory interneurons, which in turn inhibited the relay cells. Using this simple architecture, they were able to produce activity which peaked at 8 to 12 hertz corresponding to the alpha rhythm. This matched measurements they had obtained through electrodes placed in a dog's thalamus, where it was a reasonably good fit for the alpha rhythm. This model was a coupled dynamical system. Dynamical systems are used everywhere, be it weather modeling and prediction, control systems, population growth models, as well as several biological models, including neurobiological ones. To motivate an example, here are 3 equations, each of which describe the evolution of a particle in 3 coordinates x, y, and z. These are coupled equations, so the position at a given point of time in the y coordinate depends on the x, and so on. It is parameterized by three variables: sigma, rho, and beta. These three equations comprise the Lorentz system, which is used to describe and predict weather. In order to get an intuition as to what these equations are describing, here's a simulation of the position of the particle described by these three equations as it evolves as a function of the parameter rho, which is shown as r here. As r increases, the particle's trajectory follows an oscillatory pattern, and as r continues to increase, this pattern becomes richer and richer until it splits into two different orientations known as a bifurcation. As r continues to increase further, these patterns become even more detailed, reaching a point where they become completely unpredictable and chaotic. The Lorentz system is a very good example of a nonlinear dynamical system which is linear in its parameters and can produce a wide range of complex, rich behaviors. We are now going to look at a neural mass model which can produce similar sets of rich dynamics based on similar differential equations. We start with modeling the neocortex of a rodent. This is the barrel cortex, and it is called so because of the repetitive columnar barrel-like arrangement found in the cortex. The cortex has this patchy appearance in layer 4, which is one of the granular layer. It is called so because of the presence of certain specific neurons which give it this appearance. Out of the 6 layers of the cortex, layer 4 consists of granule cells, and some of these consist of these star-shaped excitatory neurons which are glutamatergic. These receive inputs from the thalamus and distant cortical areas and are referred to as spiny stellate cells. Below the granular layer, layer 5 and layer 6 are collectively referred to as the infragranular layers. These mostly consist of pyramidal neurons which have dendrites extending all the way up to layer 1. Depolarization in the dendrites of these pyramidal neurons corresponds to the output of this column of cortex. We'll refer to them with this triangle. Lastly, the top 3 layers, called the supragranular layers, largely consists of inhibitory neurons and some superficial pyramidal neurons, but we'll be focusing on the inhibitory neurons, which are specifically vasoactive intestinal polypeptide expressing GABAergic interneurons, which have the highest density in layer 2, layer 3, and we'll denote them with this red circle. So essentially, we have reduced the 6 layers of the barrel cortex down to 3 populations of neurons: inhibitory in the top 3 layers, stellate cells in layer 4, and pyramidal neurons in layer 5, layer 6. The spiny stellate cells receive input and the pyramidal cells produce the output. Let's first focus on a single population: how the spiny stellate cell population processes external inputs. The rate of change of voltage is denoted as a function of current. We use the notation with a letter and a dot on top to denote its rate of change with respect to time. The rate of change of current is modeled as a function of the present voltage, the present current, and the input. Now these are scaled by constants. These constants are the average dendritic time constant, which is tau, and h, which is the average synaptic gain. Now these are essentially changes in voltage as a function of current and changes in current as a function of voltage, current, and input. These are coupled first order differential equations, and the solution to these equations is given by this integral here, which can be equivalently expressed as a convolution. V is u, the input, convolved by an impulse response h, and the impulse response is given by h, the average synaptic gain, divided by tau times t, when e raised to multiplied by e raised to negative t by tau, where t is time. Now this is what the impulse response looks like. Tau controls the decay rate of the postsynaptic potential. The impulse response essentially maps on the output produced at the postsynaptic membrane due to a spike. So a lower tau results in a faster rise and a faster decay, whereas a larger tau results in a dampened or delayed rise and a delayed decay. Tau is determined by the distance of the apical dendrites from the soma, and this can vary anywhere between 2 to 60 milliseconds. H is the average synaptic gain, and this controls the height of the postsynaptic potential. So a smaller h will result in a diminished amplitude, whereas a larger h will reduce in a larger amplitude. To get a better idea of how the impulse response, that is the rate to potential operator, works, let's look at the convolution for different kinds of spikes. So when there is no input, there is no postsynaptic potential being produced. For a given spike, we see a postsynaptic potential corresponding to that spike. For another spike, we see an additive effect, and this happens as many spikes are present in the input. For a given burst of input spikes, we see this response which ends up saturating quickly. So essentially, the rate to potential operator converts firing rate as an input to a depolarization at the postsynaptic membrane. Now we focus on these two equations and describe how other populations are also modeled and how they are coupled with each other. For consistency with existing literature, these have subscripts. The subscript corresponds to the name of the particular population we are modeling. Note here now that the two constants, h and tau, have another subscript which is E, standing for excitatory. This indicates that the input is going to produce an excitatory response at the postsynaptic membrane. Now we add another population, the pyramidal cells, in layer 4, layer 5, layer 6. These provide inputs to the spiny stellate cells, and we include that input here, VP, referring to the present depolarization of the pyramidal cell, this is scaled by a parameter gamma 1. Gamma 1 is the intrinsic connectivity between pyramidal cells and stellate cells, and this is the average number of synaptic contacts made by the pyramidal cells on the spiny stellate cells. We also introduced another function, the S, which is the potential to rate operator. Since neurons interact with each other through discrete spikes, we need a way to convert a spike to a firing to a depolarization, and a depolarization to a firing rate. So this is what this potential to rate operator does, and it is parameterized by a sigmoid function which consists of two parameters. One is R, which determines the slope of firing rate. This essentially controls voltage sensitivity, where a smaller slope, smaller R, will result in more voltage being needed to reach the maximum firing rate, whereas a higher R will result in a steeper rise, therefore needing less voltage change to reach the highest firing rate. The other parameter is E0. This controls the maximum firing rate. So a smaller value will result in a less firing rate, and a higher value will result in a higher firing rate. Now, just a quick look at what's happening in each population. So we start with a presynaptic input, which is spikes. Now this is converted to using the rate to potential operator, which is the convolutional operator, to a postsynaptic membrane potential. So we go from spikes to a depolarization, and now this depolarization is further transformed by the potential to rate operator into an average firing rate. Now this happens in one population. When we add another population, this average firing rate is fed forward as an input, a presynaptic input, to the next population. And in this other population, the same sequence of operators apply to process and integrate various inputs and ultimately produce a depolarization, which is then transformed to a firing rate. So now coming back to this equation, now we have included the effect of the pyramidal cell depolarization, which is scaled by the average number of contacts it makes. We now add the inhibitory layer. The inhibitory layer receives pyramidal cell output, and this is excitatory in nature, and the parameter gamma 3 now captures the average number of synaptic contacts made by the pyramidal cell population onto the inhibitory interneuronal population in layer 2, layer 3. And in the equations are same, except that the inhibitory interneurons do not receive a direct input, so there is no U, but there is VP, which is the depolarization of the pyramidal cell scaled by gamma 3. The constants, excitatory constants, remain the same because the input received by the excitatory, the inhibitory interneurons are essentially excitatory in nature. Lastly, we add a bunch of equations for the pyramidal cells now. The pyramidal cells receive two kinds of inputs. It receives excitatory inputs from stellate cells, and these are scaled by the number of contacts made by the stellate cells on the pyramidal cells, captured in gamma 2. And this corresponds to these equations on the left, where we have this membrane potential of the spiny stellate cell being transformed into a firing rate, which is scaled by gamma 2. The other input received by the pyramidal cell is the inhibitory activity from the inhibitory interneurons from layer to layer 3, and these are scaled by the average number of synaptic contacts made by that population on the pyramidal cells. The equations on the right capture this. It is here that we see the inhibitory time constants and synaptic gains come in, where hi and tau I capture the dendritic time constants and synaptic gains for the inhibitory responses. The output at the pyramidal cell is essentially a difference between the two kinds of currents. So excitatory minus inhibitory gives us the net depolarization at the pyramidal cells. This is the Jansen-Rit model. It was published in 1995, and these equations describe how a system of these three populations, interconnected populations, respond to an external input. So if we have a Gaussian impulse as an input, we can look at the output of the spiny stellate cells produced by the governing equations, which looks like this. For example, the inhibitory one could look like that, and the pyramidal output could very well look like this. This is the output of the whole system. So what we have essentially modeled is one column of the cortex, which takes in an input, does some processing based on the state space equations, and produces an output, which is the depolarization at the pyramidal cells. Now, this full system has 33 parameters, which is difficult to fit and model. So it is constrained to have 5 free parameters, 2 dendritic time constants, excitatory and inhibitory. The excitatory ones are at the spiny stellate cell and the pyramidal cells, whereas the inhibitory ones are in the inhibitory interneuronal populations. There are 2 synaptic gains similar to the dendritic time constants, and lastly there is the intrinsic coupling, which is gamma 1. This corresponds to the average number of synaptic contacts. Now, based on biological constraints identified through rodent studies, the other average number of synaptic contacts, that is gamma 2, gamma 3 and gamma 4, are usually expressed as fractions of gamma 1. So for example, gamma 2 is 80% of gamma 1. So if there were 100 connections from pyramidal cells to spiny stellate cells, there would be 80 in the opposite direction. Now, this system is capable of producing very, very rigid dynamics and can model a lot of EEG, MEG phenomena. So for example, here we have excitatory dendritic time constant tau on the x-axis and the inhibitory one on the y-axis. So for different combinations of the excitatory and inhibitory ratios, this system can produce delta, theta, alpha, beta and gamma oscillations in the time domain on EEG. Now if there is extrinsic input coming to the inhibitory interneurons, then we see even richer dynamics. So for example, here in the left, there is no input to the external input to the inhibitory neurons, and the system is in an alpha oscillation. The moment we introduce some extrinsic input to the inhibitory populations, we start seeing these large amplitude spiky bursts. Now for an even larger input to the inhibitory interneuronal population, the system enters a state of high frequency, low amplitude activity. So essentially this motivates the idea that each of the populations may receive different inputs, and they can produce an even further richer set of dynamics than is possible with one cortical column alone. Based on primate studies, Feldman and Van Essen in 1991 described rules by which two cortical columns may be connected to each other. So they described three kinds of connections: bottom up, which go from one column to layer 4 of another column; top down, which go from one column to the supra and infragranular layers of another column; and lateral ones, which go from one column to all three layers of another column. These are simplified rules, and these are included in DCM as an extended version of the Jansen-Rit model. So forward connections or bottom-up connections are ones which originate from the pyramidal layer of a region and go to the spiny stellate layer of another region. Backward connections originate from the pyramidal layer of that higher region and project information back into the inhibitory interneuronal population as well as the pyramidal cell population. We can also have lateral connections for linking different regions at the same level in a hierarchy. So for example, this region on the left will produce lateral connections which go to all three layers of another region. This is usually the case for two regions which are located in different hemispheres. So there are long range projections going from one to the other via commissures. Now these lateral connections could be unidirectional or bidirectional. Now if we bring it all together, let's say we have three regions. Region 1 is a lower region and regions 2 and 3 are higher up, but they both are at the same level of a hierarchy. So region 1 receives an input, and the output via the pyramidal neurons is projected forwards to both regions via forward connections, and these connections end up in the spiny stellate cell layer. Now region 2 feeds back information to region 1 via backward connections, and which go into the inhibitory interneuronal layer and the pyramidal cell layer. Now the second region also gives lateral connections to region 3, which go in all three layers, and region 3 reciprocates these connections back to region 2, which again go to all three layers. Now, since region 2 is receiving multiple inputs, we need to update the state equations to take into account these extrinsic inputs coming from region 3 and region 1. So now we had this equation earlier where the spiny stellate cells is receiving input from a sensory stream, say thalamic input, and it is receiving inputs from the pyramidal cell population in the same cortex, and these are U and VP. Now it receives forward and lateral connections. So forward connections could come from another region, and lateral connections can come from the same column in the other hemisphere of the brain, for example. Now these two are incorporated in such a way that A of F, which is forward, and A of L, which is lateral, modulate the gain on top of the firing rate of the input from the pyramidal cells. Similarly, inhibitory neurons receive input from the pyramidal cells, which is Vp here, which is membrane potential. It is converted to firing rate by S and scaled by the average number of synaptic contacts made by the pyramidal cells on the inhibitory neurons. Now the inhibitory neurons receive backward and lateral connections from other regions. Now, so these are incorporated right here, where the backward connection and the lateral connection modulate the gain of the output of the pyramidal cells. Similarly, the pyramidal cells receive inputs from stellate cells, which comprise the excitatory activity and inhibitory activity coming from the inhibitory interneuronal population, which is in these equations here. Now they receive backward and lateral connections, both of which are modeled as excitatory gains, and they modulate the gain right here. Again, the net output remains the same, where it's the difference between the excitatory current and the inhibitory current at the pyramidal cell. This is the extended Jansen-Rit model. So it is the Jansen-Rit model plus rules for extrinsic connectivity, and these extrinsic connections modulate the gains of the pyramidal cell response generated in each of the layers. This extended Jansen-Rit model is the ERP model as it is implemented in DCM. However, this is not the only neuronal model. There are various other models in DCM. So we started with a cortical column, and we modeled 3 layers as the granular, supragranular, and infragranular layers with this connectivity. We made the implicit assumptions that there are multiple neurons in each layer, and they are lumped into a point mass. So we treat each layer, the population in each layer, as a point mean that captures activity of all neurons in that particular column. Now, instead of lumping them like this, we could also model the spatial spread of each of the layers by modeling the population as a field. In this case, we can model each of the layers as a cortically extended sheet, and this allows us to model how neurons may fire with a certain correlation. These are richer models and incorporate a lot more nonlinear behaviors. So we start with 3 populations, and we have a reduction to a point mass or neural mass models. We looked at the ERP model. There is another one called the LFP, local field potential models. This contains extra connections, extra intrinsic connections, which allow for modeling of some high frequency oscillations. Similarly, there is an SEP model for sensory evoked potentials, which contain faster dynamics compared to the ERP model. Now, if we were to not reduce to a neural mass and instead model a neural field which is spatially distended, we get the NFM model, which is the neural field model consisting of 3 populations. Now, all the models we have looked at up till now, they involved the use of a convolution operator to transform an average firing rate to an average depolarization. However, the average firing rate was not modeled as a consequence of any specific membrane properties. There is another class of models called conductance-based models, which take into account the dynamics of different kinds of receptors, for example, AMPA, GABA, and NMDA. And these ligand-gated ion channels essentially behave as conductors through which sodium and potassium and chloride ions can move through and produce electric currents. In such models, the ion channel itself is modeled as a conductor, and the membrane acts as a capacitor because it is insulating, and you have charge separated by an insulator. This RC circuit has been used commonly to model spiking dynamics, and essentially a single neuronal activity is modeled as a function of conductance and some input. In DCM, there are Morris-Lekar type differential equations which describe the evolution of such neurons in time. This is different from the Jansen-Rit model, which is a part of the convolution models. Right, so we now have another dimension, conductance based versus convolution based. And in the conductance based, there is a 3 population model, which is the ERP model, but now with conductance based Morris-Lekar type equations, which is the NMM model, the neural mass model. This was extended to focus on the NMDA dynamics later on. There are field extensions of conductance based models as well, where the spatial spread of activity is also taken into consideration, and these are the MFM and its extension with the NMDA receptor, the MFM NMDA models. Now, instead of 3 populations, if we split the deeper layer 5 pyramidal neurons into another population of superficial pyramidal neurons, which are commonly found in layer 2, layer 3, we end up with 4 populations. And again, this is rooted in a lot of biological evidence, and most notably the superficial layer of pyramidal neurons is a very good model of gamma activity in V1, for example. This microcircuit is known as the canonical micro-circuit, which consists of 2 populations of pyramidal neurons, superficial and deep, where the superficial one carries feed-forward signals and the deeper ones carries the back projections. These have conductance-based counterparts, which are the CMM and another one focused on NMDA, CMM and MDA. Right, so we've seen how we modeled a cortical column using the Jansen-Rit model, where we have 3 interacting populations. Then we added more regions and showed how extrinsic connections could interact with different populations across regions, and specifically how these extrinsic connections via forward, backward and lateral connections exert directed causal influence on the various populations in different regions. So at a mesoscopic level, we have a 1 cortical column, and we have many cortical columns in the macroscopic scale, which are now connected by long distance extrinsic projections. And at a microscopic level, using say conductance based models, we could specifically take into account more biologically constrained synaptic parameters like receptor dynamics and conductances. So this covers the section on neuronal models. We now move on to observation models. Now, as we saw earlier, the observation model takes the depolarization of pyramidal cells and translates it to observe data features using a forward model. DCM simultaneously optimizes the spatial forward model and the temporal neuronal model, which we saw earlier. The forward model, or the lead field, can be parameterized in 2 ways. The first is the ECD approach, the equivalent current dipole approach, where activity in a certain region is summarized by an equivalent dipole. This is parameterized by 3 location and 3 orientation parameters, which are optimized by DCM during fitting. The other is the imaging solution, which is a distributed solution, and it involves defining a sphere, and within this sphere of a certain radius, multiple dipoles on the mesh are constrained to the cortex. Now, the imaging solution can be more appropriate than ECD models when real source activity is in fact distributed locally within a cortical region. Also, the spatial degrees of freedom in the imaging solution can be specified and optimized using model selection. So we can fit using different differently sized spheres, and compare using model evidence which one gives us a better fit. Lastly, the imaging solution is linear in the spatial parameters, therefore results in faster inversion. Lastly, we come to model inversion. Now, the general DCM pipeline begins with defining experimental inputs, followed by selection of a neuronal model, which involves specifying how we model the cortical column, whether it's a mass model or a field model, whether it's a convolutional model or a conductance-based model, or whether we have 3 populations or 4 populations. Then we specify how, if we have multiple regions, multiple cortical columns, how are they connected, where we define forward, backward, and lateral connections. Then we pick an observation model where we choose the ECD fit or the imaging solution, or if we already have activity localized to various sources, we can choose the LFP approach. Then DCM consists of defining a likelihood model and priors, which are Gaussian and non-informative for most of the parameters. Lastly, we invert the model using an optimization process, and we investigate model evidence, which is in the form of free energy, which describes the degree of fit in terms of a balance between accuracy and complexity of the model. And lastly, we do inference based on the posterior distributions of parameters obtained after fitting. Now, the model inversion itself begins with providing observed MEG data and the specified model with priors. This is given as inputs to the EM expectation maximization algorithm, and this is an iterative fitting procedure where given the model with some parameters, it first does forward modeling and generates a response. This response is compared with the actual data, and if the fit is not good enough, the parameters are improved. Now this continues until a good enough fit is achieved, and the goodness of fit is evaluated using free energy. At the end of it, we get posterior distributions of parameters and model evidence. Now once we have fit a model, we check if the predictions of our models are adequate compared to the observed data. We test hypotheses by comparing model evidence. So this involves comparing different architectures of connectivity. Let's say whether a certain connectivity has higher evidence compared to a reduced model with certain connections disabled. This could be for an individual subject or within or between groups of subjects. And lastly, we can assess parameter estimates, either individual values or group averages, or whether significant differences exist between conditions or groups of subjects. Now, the kind of questions we can ask with DCM involve, let's say we have two candidate generative networks, one with 4 nodes and one with 3 nodes. We can ask whether network A explains the data better than network B. We can also look at whether a certain kind of connection is needed or not. So here X and Y are connected reciprocally, but in this network there is no backward connection going from X to Y. We can then see whether specific connections are modulated by experimental effects or not. We can also see whether the experimental effect is driven by extrinsic inputs between regions or intrinsic connections within each region. And lastly, we can ask which neural population in a given region or which parameters are affected by experimental effects. In summary, we have looked at the generative model of DCM, which consists of a neuronal model and an observation model. The neuronal model tells us how various populations in a specific region are interconnected and how these regions are connected to other distant regions in the brain. These neuronal models are capable of producing a rich set of dynamics in both the time and frequency domain, and they embody hidden variables that capture causal directed influences of one region on the other. The observation model takes the outputs of these different regions and translates it to observable data features on EEG and MEG. We've looked at how we can specify a model and then how inversion of this model gives us parameters that we can then make inferences about. Thank you for your time and attention.